Hi everyone, welcome to my channel Pure Biology, and today I come with another video, that is the pollen grain formation, and it is the topic related to the microsporogenesis, and it is the part of the sporogenesis. Okay, sporogenesis or the sporophyte generation. Okay, sporophyte generation, the pollen grain formation. So how the pollen grain formation occurs here? The microspores. Okay, the microspores just after their release from the tetrads are known as the pollen grain. So from the tetrads, when the microspores release, then these are known as the pollen grains. And the one of the pollen grains that consists of exine, the outer part, and intine, the inner part. This is exine is either a uniform sheath or is subdivided into outer sexine and inner nexine. So the pollen grains coating, the coat that is covered the pollen grain, this is known as the pollen wall. And this pollen wall, it has the two parts, or it consists of two parts. One is the outer part, that is called the exine, and the inner part, that is called the intine. And some cases, and this exine that is subdivided into the outer sexine and the inner nexine. And this pollen grain has apertures. Pollen grain has the apertures or the pores, and that is limited thin walled areas in the exine through which the pollen tube usually emerge during the germination process. So what is saying that? There is a pore in the pollen grain, and the pollen grain's pore is limited which region? The thin walled areas of the exine. Thin walled areas of the exine, there is the pores are seen, and these pores, through these pores, the pollen tubes that arises and that emerge and during the germination. Okay, during germination process, this pollen tube that arises from where? From the pores that remain in the thin walled areas of the pollen grain. Next, that this allows the grain to change in volume during the changes in humidity. So which allows means here the pollen tube, this allows the pollen grain to change in volume during the changes in humidity. With the changing of humidity, the volume of the pollen grain changes due to the formation of the pollen tube. Now the aperture, and here also important role is played by the aperture. The apertures may be rounded or furrow-like. If the aperture or the pores that is rounded in shape, then it is called the porate pollen. And if it is the furrow-like, or the pores just look like the furrow, then it is called the colpate pollen. The colpate pollen is known as the, according to the structure of the aperture. And their numbers also varies, and the basic numbers in angiosperms, that are 1. If 1, then it is called the monocolpate pollen. That means which one is the one? The number of the pores. If the pore number is 1, then this pollen is known as the monocolpate pollen, and this is seen in the monocotyledons. And if the number of the pores that 3, then it is called the tricolpate pollen, and this is seen in the dicotyledons. Clear? And the most distinctive chemical component of the pollen wall in the is the sporopollenin. Okay, sporopollenin, and that consists of oxidative polymers of the carotenoids and the carotenoid esters. So one chemical component that is also present in the pollen wall, and that is known as the sporopollenin, and this consists of oxidative polymers of which carotenoids and the carotenoid esters. So the chemical name is the sporopollenin, and that consists of the carotenoids and the carotenoid esters polymers, and these are the oxidative polymers. Now the sporopollenin is resistant to various chemicals. Okay, it is made up of in such components of the constituents that it resistant due to presence of this chemical, the pollen grain that is resistant to the various chemicals. And the pollen wall, that development begins while the tetrads are still enclosed in the callus. This callus, I had a mistake in my previous video, that is related to the microsporogenesis, that I mentioned there the cellulose, but not cellulose, that will be the callus. Okay, so within the callus, the pollen wall development that begins while the tetrads are remained within the callus, and but because initially they haven't any wall. Okay, slowly or gradually the pollen wall develops. Clear? So this is the structure of the pollen grain, and now, or you can say the structure of the pollen grain, or the how the microspores that mature to become the pollen grains. And this is the second part, that is the megasporogenesis, and here the space is short, so the continuation part I have to write here. So just I rub this one and write the next continuation part of this megasporogenesis. Okay, so continue with my channel. So here the megasporogenesis is what? Megasporogenesis is the process by which the megaspores are formed in the megasporangium. Okay, due to the meiotic divisions of the megaspore mother cells or the megasporocytes. This is actually the megaspore mother cell, known as also megasporocytes. Clear? Meiotic divisions of the megasporocytes, the megaspores are formed. So what is the megasporogenesis? The megaspore formation in the megasporangium, and it is formed due to the meiotic divisions of the megasporocytes. Clear? And here the linear tetrad of the haploid megaspores are formed. Here also tetrad formed, megaspores tetrad are formed. That means 4. Tetrad means 4. 4 megaspores are formed, and within 4 the 3 degenerate. Only 1 remaining functional. Clear? And which lies where? This is lied to the chalazal end, or in this situation, when the remaining one or the functional one that remain in the chalazal end, then this type of megaspore is known as the polygonum type. And the ovule developing from the placenta of ovary is the site of megaspore formation. So which is the site of the megaspore formation? Where the ovule developing from the placenta, the place from where the ovule developing from the placenta of ovary, this is the site of megaspore formation. And obviously embryo sac is formed. Embryo sac is formed, and which is known as the female gametophyte. And this ovule consists of nucellus central body with vegetative cells that enclosing in the sporogenous cells. Clear? So what are the part? Nucellus central body with the vegetative cells, and which enclosing in the sporogenous cells. Clear? And here 1 or 2 integuments are there. If one, this is known as the unitegamic, and 2 integuments are there, this is known as the bigamic. Clear? And the stalk, this is the stalk, basically. This is the stalk. The stalk connecting ovule to placenta, and there is a stalk that connecting the ovule to the placenta. And the region where the nucellus, integuments and funiculus that merge, known as the chalaza. Okay, that funiculus is another part, and obviously nucleus and the funiculus, the two parts are there that enclosed within the integuments. And where the chalaza are formed, the nucellus, integuments and the funiculus were merged. This portion is known as the chalaza. And now the micropyle, this is the opening, and the micropyle remains where the inner integument arch over the nucellus. So where this is micropyle is formed, the opening, and micropyle is the opening, and it is formed where the inner integument that arch over the nucellus, that is bending over the nucellus, and there is the micropyle or an opening is formed. One or both integuments that involved in the formation of the micropyle. So if one integument that is necessary for the micropyle formation, or sometimes the two integuments both are involved in the micropyle formation. And the ovule with the large nucellus, this is known as the crassinucellate. Okay, crassinucellate ovule. And one with the small nucellus, this is called the tenuinucellate. Okay, so there are two type of ovules are seen. The development of the archisporium that starts in the nucellus. So within the nucellus, the development of the archisporium that starts, and a single hypodermal cell within the nucellus that become enlarged and modified to form the primary archisporial cell. And this archisporial cell is important. Why? Because this divides and to form the primary parietal cell and the primary sporogenous cell, and which function as the megaspore mother cell that produces the megaspores through the meiosis process or meiotic division. Clear? So why this primary sporogenous cell is important? Because this function as the megaspore mother cell, and it produces the megaspore through the meiotic division. Clear? And here important thing is that the cell division is occurring in the periclinal division or periclinal pathway, or periclinal division is known means that the two types of division generally seen. One is the periclinal, another is the anticlinal. So this is the horizontal and the vertical division mentioned here. So the division of the cell division here seen only in the periclinal division, not the anticlinal division. Clear? So in that way the megaspores are formed. Clear? So megasporogenesis and pollen grain both are explained here. So I think you understood all this. And if you are the newcomer, so please do share, subscribe my channel if this is helpful to you, and hit the bell icon for getting the notifications. Okay, thank you.